All right. Hi, everyone. It's nice to be joining you. My name is Saskia Vries. I'm speaking on behalf of myself and David, who's in the back. Nobody turned around now. There he is. And I'm going to be talking to you about some aspects about the data platform that we've been developing here at the Institute for Neural Dynamics, and kind of how we use this in order to organize our data, how we use this to collaborate and build a platform for science. So just very briefly, some of the motivation for why we need to have some robust data solutions for neuroscience today. And I think probably many of you are aware of some of these issues, and that's probably why you're actually here at this workshop. One of the key things happening in our field is just the explosion of the amount of data that we're able to generate. And this is both because there's just hundreds and thousands of us that are doing research, but also because the techniques that we're using are generating much more data than they have historically, right? This is the Stevenson courting plot showing you the number of simultaneously recorded neurons as a function of publication date. And you can see that, you know, we went from doing our single cell recordings with tungsten electrodes back in the 50s and 60s, and now we're looking at large thousands of neurons at a time simultaneously recording. And that's just in one experiment, and then we can churn out experiments day after day after day. And these data sets are large, and there's lots of these data sets. And one of the things that we're finding is as we scale the data, it's, you know, the title of this review article is that it calls for new insights. We're finding that a lot of our ideas and theories about the brain are coming into question because of having larger, more multidimensional data. And it also means that, you know, what it takes to kind of really dig into this data and understand it is also a new thing. When Hubel and Wiesel were recording receptive fields with their tungsten electrode, they literally were just like listening to the electrode and drawing X's and triangles on the whiteboard. I don't know if you guys are familiar with these videos, but like that was their analysis, was like just like listening in and kind of like estimating. And that doesn't scale in the way that we need our analysis to scale. And so it requires not just that we have to handle large volumes of data, but we need to bring in people with different areas of expertise who can collaborate to generate the data, but also interpret the data and analyze it. The other component of what's happening in our field is just the degree to which our experiments are becoming more multimodal. And this is a review paper that we published here at the Allen Institute that summarizes some of the work of the past decade in the Allen Institute for Brain Science. But just generally in our field, we're finding that there's just more and more ways that we can combine information from different modalities of data in order to really gain insight into circuitry and function of the brain. But that means, again, we need to be able to draw different people, different areas of expertises, and different modalities of data to bear on one another. And so this is a lot of the motivation for some of the key purposes and goals of the data platform that we've been building in the Institute for Neural Dynamics. And just to kind of go over some of the high-level goals, we want to be able to support and maintain standardized and reproducible workflows and pipelines. We need to be able to handle large volumes of data using best-in-class methods and be able to do so reliably, reproducibly. We need to enable multimodal experiments and analyses, and this means building up modular workflows. If we have pipelines for a number of different modalities, say behavior videos, extracellular electrophysiology, we need to be able to, you know, process these different streams of data, but then also bring that information together effectively and be able to pull kind of the key information in a reliable way. Our platform needs to be designed for flexibility because a lot of times our experiments expand and grow and change as the science leads us in new directions. And often when we build pipelines for a very specific experiment, as that experiment changes, the pipeline is going to become brittle and break as well. And so we've been building this platform in order to anticipate that we're going to want to add new things in. We're going to want to change behaviors. We're going to want to add different types of optical probes and things like this as these technologies continue to develop. And likewise, on the analysis side, we need to be able to, we anticipate being able to bring different types of analyses and approaches to the data. And then finally, we need to facilitate collaboration and open science. We want we know that we need to integrate different people's expertises in our work, and so we need to build a platform that allows people to access and work with the data effectively, and minimizes barriers for sharing data, sharing code, making that work reliably and reproducibly, and not just among our friends and family, but throughout the broader community. So this is a challenge, and this type of collaboration is a challenge even when we're all in the same building, even in a building like this. Like we can have 3 people that are working together. They do different types of things. One of them is doing a blurry electrophysiology experiment. Another is studying morphology. The third develops analytical models. They're really excited about this project. They sit in desks on the same floor of this building, and yet they have a lot of data that they need to share with one another. They need to move that data back and forth. And moving data becomes a really big challenge, and it's a challenge in a building like this. It's a bigger challenge when we're trying to collaborate with somebody across town or across the country or internationally. And so we've taken an approach of putting all of our data and compute onto the cloud, and so that we're bringing people to the data rather than moving the data around. This makes it really easy so that these 3 people that are all working with this data are always using the same data. It's always the same copy. There's not a version 2 final. No, really, this is the final edit of our spike sorting, trust me. It's always the same thing that they have access to, and they're able to work with it in the same environment. This is great for facilitating our own collaboration in the building. It makes it even easier when we're collaborating with people outside of our building, because again, that data movement problem is much harder when we're starting to talk about people that aren't in the same system on the same network, and then allows us also to open up these resources and make them available to the broader community. And so this makes it so that the same platform that we are using to collect our data and process our data, we can now share with our collaborators and make accessible to the scientific community in order to make a shared, open, collaborative platform. So what does this look like? What are kind of the key components? First, this platform means that every part of this workflow needs to be accessible and openly available. This includes both the raw data, the primary data assets, as well as derived results. So the output of our spike sorting or our cell segmentation. We need the analysis code. All of the metadata and protocols that go with our data need to be accessible. We need everything to be standardized. Our data is stored is immutable and conforms to standards that have been agreed upon in the field. We want things to be transparent. This is both the data and the code need to be both accessible but also well documented so that it facilitates reuse. And finally, we need a platform that supports reproducible analysis, which we do through containerized compute that allows us to have version-controlled pipelines, and it allows us also to keep track of the provenance of our data assets. So how we implement this, so I've already mentioned that we're doing all of this work on the cloud. Our data goes into an open bucket on the AWS. And we make our data, we put our data on the open bucket shortly after we actually collect it, after we acquire it. We upload it with metadata. All of our data and all of our pipelines and analysis are synced into repos on GitHub, and so those are also public that anyone can access and see. And then we have protocols. You've probably already seen the protocols, some of them from the workshop, but when we're doing the experiments, we're keeping track of the protocols that we're doing. Those are published protocols on protocols.io that anyone can access and even use. All of our data is being put into standardized data formats. And like I said before, we really lean into these community standards. We use Neurodata Without Borders for our physiology and behavior data, and OME-ZAR for our light sheet imaging data. We've also been developing a metadata standard that I'll tell you more about that we use to document these data, and we use all of these kind of consistently across all of the data that we have. We make things transparent through our metadata schema. Each data asset that we create, whether it's through acquisition or through data processing, is documented by a metadata file that accompanies it. And this goes into a metadata catalog, a JSON document database that we can then use to easily query the data, and we can use that to find important information about how experiments were done or how derived results were created. We also develop different types of data portals for visualization and reporting. I'll show you a little bit about this as well. Finally, the reproducible analysis, I think, is one of the really key things, and I want to talk a little bit about this. In order to have reproducible analysis, we lean into a platform called CodeOcean, which is a cloud-based data analytics platform that's built around this concept of containerized compute. So let me talk about why this is important. So a couple of the key things when it comes to reproducibility and provenance is being able to understand how our data was generated. So when it comes to the acquisition of data, this means details about what methods we used, what were what were the reagents, what lot numbers did we use, where did we place our electrodes, which coordinates did we put them in. And this all falls into this this idea of scientific rigor, is to be able to have this really clearly documented. When it comes to processing data and analyzing data, we also want to keep track of how did we do that? Like what version of the code did we run? What were the parameters set at when we did our spike sorting yesterday? Because I don't like the results of that. I'm going to run it again, and I'm going to run it with a different version of the code, right? And so keeping track of what version of code, what parameters were set in order to get our derived results. Having reproducible analysis also means that we can develop shared tools and that we can trust that they're going to run the same way every time we use them. And so if I develop a pipeline for cell segmentation, and you're going to use that as well, it's going to work the same way for you as it does for me. And so that means that we can actually trust the outputs that we're generating, which becomes a really crucial thing for when we're trying to collaborate with one another and communicate about how we're analyzing our data. And then finally, one of the important things for reproducibility is having full access to the full workflow. I think oftentimes when we share data, we're sharing highly derived results, often not just the output of spike sorting, but the output of, now I've created PSTHs and I've computed this obscure metric, and here's a table of all these metrics, which can be useful, it can be valuable. I don't want to undermine that, but we aren't able to really understand exactly how you got to those results or recreate that with only having kind of few snapshots along the way. So having full access to the full workflow allows other users to be able to understand not only how it was done, but to be able to enter into it at different parts. So what this looks like, kind of to underscore why this is important, if you are doing, say, calcium imaging, if you're reading a paper on calcium imaging, you'll probably see a sentence like this in the methods. We use Sweet2P to process our data. This is a fine sentence to have in your methods. It's true. It's good, well done. But it's a little bit like these instructions on how to draw an owl, because it really doesn't give you the full picture, right? Like, yes, that is the tool that you use, and it's a good tool, but there's a lot of things that are going on in that sentence that aren't being communicated, right? And exactly how you configured Sweet2P, exactly what parameters you used, all of that is missing. And so what we're trying to do is kind of create the full owl instead of just the two circles in order to describe how and to communicate how data has been derived. So this means having all of the data, having the parameters, keeping track of kind of all of these different steps. Oftentimes we kind of ignore a lot of the pre and post processing steps that become really crucial for determining how that standardized pipeline actually performs on a particular data set. Another important thing about it, though, is that it needs to be able to run anywhere. And so again, if I have developed a pipeline that works well for me and you want to use it in your lab, it needs to be able to work on my system as well as your system. So let me talk a little bit about CodeOcean and some of the features that it has that we have found to be beneficial for a lot of our data work that we're doing in neural dynamics. So CodeOcean is built around this concept of containerized compute based on capsules. These capsules containerize data, code, and the environment. So each capsule has, we attach specific data assets to it. It has code that is synced to a GitHub repository, but it has specific code. And we also will set the environment for that capsule. And what that does is if I now can share this capsule with somebody else in the CodeOcean environment, and because the environment is set in the capsule, when they run that code on that data asset, they're guaranteed to get the same result, right? Now, none of these things are like highly unique, right? Code is synced through GitHub. Our environment is set with a Docker file. But one of the things that's nice about CodeOcean is that it lowers the barrier on using Git and Docker and things like that, so that many of our scientists who, have like an understanding of GitHub and like understanding of Docker are able to use these like appropriately, right? And so the way what this means is, in fact, there's like a GUI for setting your environment. You say, I want to add matplotlib and pandas and NumPy, and boom, it's pinning particular versions, and you've got a Docker file. And I never have to open up the Docker file and write it myself, right? And I can share that with somebody else who has never heard of Docker, and they're able to run it and have the environment configure appropriately. Another great thing about CodeOcean is that when we use a capsule like this to analyze a data asset and we save some results from that, those results then have a provenance map that shows where they came from, right? So this is a derived result here, and what it tells me is what the raw data was that went into it and what capsules were run in order to create it. And I can actually go in and interact with this and see when I look at this capsule, it'll even show me kind of what parameters were set or what other features of that capsule there were that yielded this particular result. And so it automatically tracks the provenance of our derived assets. And then finally, we can use this to build pipelines by tethering together different capsules that do different things in order to build up a larger workflow. One of the great things about this, and so we use this, and you'll hear more about this later, to develop our spike sorting and cell segmentation pipelines. Each capsule has its own environment, and so some environments might be more complex than others. There might be packages that are incompatible, and so we can separate those processes into distinct capsules. Some of these steps might require a lot of compute, and because we're doing this on the cloud, that compute costs money. And so we can put only the expensive things in the things that require heavy compute, and then do the more lightweight analyses can be in capsules that have fewer compute resources attached to them. And this allows us to make this a more efficient pipeline. So we can string together these capsules. This then creates a reproducible pipeline that works, again, in the same way as each individual capsule, and that everything is specified in terms of the environment and all of the components. All right, so as I mentioned, so these pipelines, I've been emphasizing them in terms of how they fit into kind of the reproducible aspect of our data platform. By having these containerized workflows. But because each of these then is synced with our GitHub, they also are completely transparent. So you can go onto GitHub, you could scan these QR codes if you want, and you can go and find all of these different pipelines. And not only are they visible and, sorry, go the other way. Not only are they something that you can go and look at the code, but you can deploy these in your own local environment, right? So we've designed them and we use them on CodeOcean, but these are in essence a Nextflow script that can be deployed on your local machine or on your local cluster at your university, and so it doesn't rely, they're not dependent on being in CodeOcean to be able to use the actual pipelines that we've developed. But again, CodeOcean allows us to develop them pretty easily and lowers the barrier for our engineers and our scientists to build them and to maintain them. All right, so you'll hear more, I think, from Ariel after me, who's going to talk about some of the pipelines that we have. But we have a handful of pipelines that we've built in this system for handling data from EFIS data, some of our imaging data, behavior videos, calcium imaging, and fiber photometry. And all of these are available through our GitHub. All right, so I want to shift gears to talk about metadata and file standards and why these are important. So standard files are really important to make data accessible and usable. If I'm sharing data with you, you want to kind of understand how to open up this file, find things in it, and use them. And the more that we use shared tools, shared formats allows us to build shared tools on top of those formats and in order to facilitate this. And as I mentioned earlier, there are a lot of good community standards like NWB and OME, and these really excel in their data organization. but they usually fall short on how they document the data in the types of metadata that they require and facilitate. And so this is why we've been developing our own metadata schema, because rich metadata is really crucial for communicating to each other about what data is. And this is important when we're collaborating within a team as well as outside of a team to facilitate that data reuse. So just real briefly to illustrate this, those of you who are kind of familiar with open science have probably heard about the FAIR principles about data being findable, accessible, interoperable, and reusable. And if you go and look at kind of the paper that introduces these principles, one thing that you'll notice is that all of these principles really come down to having rich metadata to document data. So making data findable and accessible, interoperable, reusable, all of these come down to things. I've highlighted the word metadata in each of the sub-points about them. The only two that don't have metadata talk about the protocol, and I would argue that protocols are just part of metadata. And so really that becomes kind of the linchpin of open science. This is a recent study that I think kind of illustrates this. This is a study on replicating some various AI results where they took a couple of 30 different studies, and one of the things, and to see how well they could reproduce the results of them, and one of the things that they point out in the abstract is the quality of the data documentation correlated with successful replication, right? So the more they understood, the more documentation there was on the data, the better they were able to reproduce the results of the analysis. And then finally, the key thing, and I've kind of already said this, but in order to collaborate, whether that's with your teammate who you sit next to in the office, or with somebody that you only talk to once a year at conferences, or with somebody that you've never met, in order to facilitate the type of multimodal type of collaborations that a lot of the work that we're doing now depends on, really means that we have to have good documentation and document it in a way that can be understood across different disciplines and modalities. So we've been developing this schema. I'll just briefly kind of tell you about some of the things that go into this. Each data asset that we acquire, we document it with information about the subject, so the animal that the data came from. For us, this is almost universally mice, but it could be any type of animal. We keep track of other information about the animal, such as its date of birth, its sex, its genotype. We keep track of some housing information and breeding information for the animals that we breed here at the Institute and house, and other things about, like how they're housed, what their life cycle is, things like that. We then keep track of any procedures that are done to the animal or to tissue derived from the animal prior to data collection. So this could include, in this example, some stereotactic surgeries where we injected a virus, and we want to keep track of where we injected the virus, what was the plasmid of that virus, what was the lot number of the prep lot that we used. This mouse also has fiber optics implanted in it, and again, we want to know where those fiber optics are positioned, as well as some of the optical details of those optics. And I think this animal had some water restriction, and so when did that start? What was the protocol for water restriction? We also keep track of instruments that are used to collect data. So for this animal with fiber photometry, there might be some lasers involved. There might be some behavioral cameras, some behavioral apparatuses that are being used. And then during data acquisition, we want to know what was the stimulus presented, what were the behavioral parameters that were used, how was the fiber optic configured when we actually were acquiring that data. And other things that might go into we want to keep track of both the data that's being collected and then any behavior or stimulus that's being applied. Then after we've collected raw data, there's always going to be some processing that happens. This might be very minimal, or it might be very elaborate, right? We might be doing a full spike sorting type of thing. But so again, we keep track of the processing. I've talked about kind of that provenance tracking, what code was used, what were the parameters, when was it run. And so this will describe each of our assets. We have two other pieces. One is kind of a high-level data description. This is more administrative, but what project is this a part of? Who's responsible for it? Where was the funding from? What's the license of its reuse? And then finally, an important piece is the quality control of the data. And this is actually kind of the only piece of our metadata that can be updated and added to after the data is acquired, because we can evaluate quality control kind of during different stages of acquisition and processing and analysis. And I think you'll hear more about that from Dan in a few minutes. So this is one of the assets, one type of experiment. One of the things you probably know about us at the Allen Institute for Neural Dynamics is we do a lot of different types of experiments. And so this top one that I showed you is an example of a fiber photometry experiment. But for instance, we also have a bunch of light sheet experiments where we have very different information that we need to keep track of, because we, instead of any live animal experiments, we perfuse the brain, we expand the brain, we use antibodies, we use fancy microscopes. And so our schema has been developed in order to really accommodate a wide range of different types of experiments within a consistent, standardized schema. And one of the things that we lean into is trying to use persistent identifiers that come from public databases whenever it's possible, in order to describe a lot of the components that go into these experiments. So persistent identifiers are things that tell us, so for instance, when we're using transgenic mice, there's a mouse genome informatics database that documents different types of transgenes. And so I can be really precise, because there might be 5 different aliases for the same transgene. And so when I call it one thing and you think of it as another thing, we might not understand we're talking about the same transgenic line. This allows us to put an identifier on it to be very precise, so that we know we're talking about the same thing. And so these are a few of the ontologies and databases that we lean into, but there's definitely more as well. So each experiment, each data asset has this type of metadata. Each asset has a JSON file that consolidates all of this information. These JSON files go into a document database, which allows us then to, we have all of the metadata about all of our data in one place, and then we can create, we can query this metadata when we're looking for particular assets, or when we need information about specific assets. So our software scientific computing team has developed kind of a ecosystem of tools around this. I won't really go into a ton of details. We have some tools for ingesting metadata. This is one of the big challenges, is how do we get all of these details into our records? And this is, one of those overwhelming things, because you're like, I really don't want to sit and type all of these fields every single day. And so we've been developing a variety of tools that can automatically grab this during experiments, or from, say, like our breeding database and things like that. But we also have, I mentioned the QC, we have some tools for annotating the data, as well as tools for querying our database. So the QC portal, and again, I think you're going to hear more about this, but this is a tool that allows users to visualize kind of snapshots of the data, both raw data and processed data, and particular metrics that scientists have decided that they want to evaluate. And so this allows like an actual interface for scientists to go in and say, this is a good, this aspect of the data is good, that's bad. We evaluate each modality independently, and so you can have an asset where the EFIS recording is great and the behavior videos are terrible. So depending on your analysis, if you don't need the behavior videos and you just need good spikes from a particular region, maybe you want to use it. But if you do need the behavior videos, you maybe want to skip that one. When it comes to querying the doc database, this is big JSON files. We can use MongoDB to write queries. Usually when I say this, nobody did this right now, but usually when I say this, everybody's eyes kind of glaze over, because they're like, oh gosh, that's another tool I need to learn how to use. On the one hand, it's actually not that complicated. You've probably done some sort of pandas query or something like that. It's the same type of idea. But the other thing that I'll tell you is that we've also, we can build up these types of queries off of the database, but we've been developing a chatbot that will generate these queries for you based off of natural language. And so a member of my team has developed this chatbot that will query the database. It will query the records in order to summarize them or pull specific features out of it, or even kind of pull kind of requirements of the schema just out of the schema requirements itself. So a couple of examples of what this can look like. Here's a query of what are all the brain structures that were injected in the thalamus in the middle project. And it does some work, and it comes up, it returns this answer. It says here are the 10 regions where we injected that were injected in this particular project. And it also returns the code that will create that query on the database. And so you can take this code, you can put it into your own workflow if you want to. It allows you to also validate it. So we do a lot of benchmarking on this. Another use of the chatbot is, this query is tell me about mouse 747107, and it gives me a summary of this animal. It's a female mouse with the genotype of such and such. It underwent planar optical physiology using the mesoscope setup, the 920 wavelength and area Viz-P at multiple imaging depths, yada, yada, yada. And like I've written half my methods section, right, just by pulling up these metadata records. We've also, this is, really fresh off the press, but this chatbot in this format is like a chatbot I go to a specific website, and so when it spits out code like this, I can copy that code and take it into a different workflow and use it. But we've now that we've got these MCP, these model context protocols, we can actually integrate this directly into our analysis work. And so this is a capsule on CodeOcean where, and I'll just tell, I can't demonstrate it right here, but basically we said attach, find an asset of this project, attach it to the capsule, open up the NWB file, and we asked it to create a bunch of visualizations. This is one, it's from a calcium imaging experiment where they had a neuron that they photostimulated, and so make a plot of all of the ROIs with the stimulated ROI indicated, and the chatbot, attached the data, created the capsule, got it running, wrote all this code, made these visualizations. So being able to integrate all of this information together directly into the workflow. All right, so just to summarize and coming back to kind of these key principles of our data, of our platform, making the data accessible and transparent and creating these reproducible tools and doing so in a shared environment. And really the key motivation for why we've been investing a lot of this energy into doing this is that we want to move away from a situation where we're generating a lot of data that we then kind of disseminate out into the world and kind of, whatever happens to it, but rather to build this platform that enables us to draw in our users and collaborators throughout the community so that we can all kind of work together to solve some of these challenges that we as a field continue to work on and continue to face as we work with our data. All right, so with that, I will wrap up. I know we're probably running behind, so thank you.